Maybe here we can just like extend and we can talk about your approach where you use DNA nanotechnology. So interestingly, all the life forms, they use DNA as an information, right? This is what we know, at least so far. But you have decided to use DNA not only as an information storage machine, but also for the building blocks of a cell, right? So what is your approach? How you are exploring DNA nanotechnology?
Mm-hmm. So, I mean, first of all, I should make it very clear. I think there's no reason whatsoever to be dogmatic about your choice of hardware, so to say. So, DNA nanotechnology seem to be useful for us for the moment. It's also, what we can do well. But I think it's also part of the fun, and it's really necessary to keep scanning the horizon for new tools as they come up, right? Like there's no reason not to use 3D printers to make synthetic cells. We've played around with those, right? There's no reason not to use, protein engineering, RNA nanotechnology, whatever new tools may pop up. Directed evolution is an important one, microfluidics maybe, So whatever new tools pop up, I think let's just be open because our task is complex enough. So make our lives as easy as possible. For me, DNA nanotechnology is quite useful because it, first of all, as I said, like DNA is capable of encoding information, but it's also capable of carrying out certain functions. And we were able to build functional architectures like DNA nanopores, like mimics of DNA cytoskeletons, like DNA structures which cause fusion of vesicles, the structures which can shape vesicles, so can alter the phenotype of our synthetic cells, so to say. So this is why we learn quite a lot from interfacing DNA nanostructures, functional DNA nanostructures with membranes. The other reason to do this is simply because while we go towards our dream of building a synthetic cell, we also build tools that are actually useful for cell biology, right? So if you can remodel a vesicle with a DNA nanostructure, you can likely also remodel a cell with a DNA nanostructure. And then you can start to ask questions, how do certain membrane properties like membrane curvature influence processes like endocytosis or...
So I think DNA nanotechnology is certainly one tool out of many, right? It's part of a larger toolbox. And the reason I find it useful is because you can build components for synthetic cells, but you can also directly use these components inside living cells as biophysical tools, right? So think about, for instance, questions like, how does cell-cell adhesion impact collective cell migration? We can use the same linkers that we use to link our synthetic cells or lipid vesicles. We can use them to link cells and, engineer their binding strength and so on, which is something that would be actually quite complex to do with protein engineering. So all the tools that we build towards our big vision of building a cell, they're directly applicable as biophysical tools in biology. So this, for me, is another reason to keep developing these kind of tools. And yeah, as I said, DNA can encode information, but it can also be functional. Even more so, I would say this is true for RNA, because right now I have to say one big flaw of our systems is that quite often we need chemical functionalization for function, right? So for instance, for membrane spanning DNA pores, you need to attach cholesterol tags in order to make them self-assemble into lipid membranes. Or if you build a cytoskeleton, and as we did, and you want to assemble it in a reversible way using DNA, we had to put azobenzene as a photoresponsive molecule into the DNA duplex. And this is, of course, chemically challenging. It's a synthesis that can be done, but it cannot be done inside a living cell or inside a synthetic cell, and probably never will be, So this means that we would, of course, love to genetically encode our structures, also because then we can start to use directed evolution to make them better, or use directed evolution of vesicles to improve the function. But, if you require crazy thermal annealing, or if you require purification, or if you require, in particular, chemical functionalization for function, then this is not really compatible with the chemistries or with the environment that you have inside of a synthetic cell. And so this is why I think RNA origami, RNA nanotechnology is even better. So what we are now trying to do is kind of transfer the knowledge that we gained already working with DNA also to RNA structures.
Yeah, this is exciting. And so what do you mean by DNA or RNA origami?
So DNA, let's start with DNA origami. So DNA origami, or the field, DNA origami is a subset of the field of DNA nanotechnology. DNA nanotechnology is really about repurposing DNA as a construction material. So taking it out of its context from genetics and using it purely for architecture. And this is based on the famous realization by Nat Seidman, who realized in the 80s actually that you can use nucleic acids to build stable nucleic acid junctions. So anecdotally, this was when he walked past one of the Escher's paintings that showed these kind of fish in a crystal-like arrangement. He walked past this painting and thought, what if we replace the fish with DNA double helices? And, you know, this is what he did. He envisioned that one would be able to use this for protein crystallography. So he was actually a structural biologist who was interested in crystallography of proteins. And as we know, some proteins are challenging to crystallize. He thought, okay, what if we actually place them in a crystal lattice built from DNA? And this is when the field of DNA nanotechnology was born. And sadly, Nat Seidman passed away some time ago. And, you know, we really have to give him full thanks for establishing such a collaborative, such a, you know, thriving field in science that, you know, is all based on these really brilliant ideas, I think. So that's the field of DNA nanotechnology, is about constructing with DNA. And then I would say it really took off or kicked off when Paul Rothman developed the DNA origami technique, which is basically a specific version of DNA nanotechnology where you take a long single strand of DNA. It's actually taken from a virus, a viral genome, or the genome of a bacteriophage, the M13 phage quite often. It's a long single strand of DNA that you can isolate. It's got 7,000 to 8,000 bases. And then basically you take this genome and you synthesize short synthetic oligonucleotides of maybe 50 bases or so that can bind the long single strand of DNA in specific positions. So imagine you've got a long single strand of DNA, you sequence it, and one end has a certain sequence, the other end has another sequence. So now you can envision designing a short piece of DNA which is complementary at one end and complementary at the other end. And then you pull your long single strand together in a loop. And now if you take more of these short pieces, you can start to fold the long piece into more complex shapes. And like the famous smiley or other DNA origami shapes that have been made since. So DNA origami, really with 200 short strands in this viral genome, you can build finite sized objects on the nanoscale. And the beauty is that each and every of these 200 oligonucleotides has a unique sequence and a unique position within the DNA origami. And this means you can use DNA origami also as a molecular breadboard to position other components, you know, be it fluorophores. So you can use this for standards for super resolution microscopy, for instance, or be it antigens to position antigens at a given spacing or valency to improve immune response or to probe immune response. So there is really a vast variety of applications that the DNA origami technique has by now. And synthetic biology is certainly one of them, an interesting one to try and, you know, engineer molecular hardware for synthetic cells. Yeah. And RNA, so that's DNA origami, right? RNA origami, you could naively think, well, it should just work the same way, right? Also, RNA can base pair, and why not just take a RNA strand and fold it like you would fold DNA based on canonical basis, so to say, based on, you know, A binds C, A binds T and C binds G, right? But actually RNA is much more flexible in terms of the structural motifs that you have at hand. RNA can form kissing loops, RNA can form dovetails. So actually the design paradigms with RNA are actually quite different from DNA. This is what we are learning right now. And the beauty of RNA is you can design the structure such that they fold a bit more like proteins do, right? With DNA origami, what we do is we design the sequences, we put them all in a pot, we heat them to 80 degrees, we cool them down slowly so that they can basically thermally anneal to find their energy minimum. With RNA, the beauty is that you can fold an RNA origami from a single strand of RNA. So basically more like a protein would fold from a single chain of amino acids. You can also fold an RNA origami from a single chain of nucleotides, so to say, making use of this also non-canonical structures like dovetails or kissing loops. And this means that RNA origami can really fold in a co-transcriptional manner, just like proteins fold co-translationally. And this is really beautiful work by the group of Elbe Anderson and others who worked on this co-transcriptional folding. And I think this will open up a myriad of new opportunities to really design and genetically encode functional structures also for synthetic cells.
Yeah, and
it is really new. I have no idea.
Yes.
We're just starting to explore it. So, don't quote me on this, but we'll see in a few years, I guess.
Yeah, I mean that's fine, and I mean that's the exciting part, because I'm totally with you in terms of when it comes to like what we can engineer to like and what we can incorporate in synthetic cells, for example, because definitely nature has explored certain chemistry, but that doesn't mean that's the only chemical space that we need to stick to. I mean, the kind of systems that Stephen Benner is exploring or Lee Cronin is exploring, that they want to go really at the basic level and just try out with any possible chemistry and see if we can kind of get what you said, life 3.0 or 6.0 or whatever. So
And I don't know, like I think Kate was saying that, first we have to work with what we have, which is in a way, it's a fair assumption to say, okay, like we kickstart our systems by using what is there already. We make our lives easier. We make a life 2.0 that works kind of like life 1.0 does, and then we go from there and get crazy. And and the other way is to say, no, actually, we may approach it from the other side and think, okay, how simple of a solution can we imagine? And this may not be the life that the solution that life came up with, right? Like evolution is not designed to make something simple. I mean, the life that we have is extremely robust. One really has to say this, right? It's solutions exist for the most diverse environments, right? So maybe and this is also, by the way, an interesting lesson from systems engineering, that every system is part of a larger system. So of course we cannot think of life without thinking of its environment. Every living system has to function in a given environment. So I mean, we need essential amino acids. We cannot live without food, and we cannot produce everything with our own bodies, right? Other types of cells are maybe a bit simpler. They need maybe even their lipids and cannot produce all of their lipids, like mycoplasma, right? You need to feed them certain lipids so they kind of act as parasites, right? Parasites need their hosts to work. Viruses even more so. Like they need a whole host cell and to basically capitalize on their machinery. So I would say, maybe our idea in synthetic biology of making everything autonomous is also a little bit too ambitious, at least for the beginning. So I would say in the beginning, our synthetic cells will really be as simple as possible and function only in an extremely resource-rich environment where we resupply all the reagents that are needed at the right time, so to say. So we are kind of helping to kickstart this process of evolution. But then once we are there, we hope that evolution can take care of actually maybe getting rid of some of the components and, increase the degree of liveliness or decrease the degree of complexity of the environment, right? So it's also an interesting concept, I think, which is getting more and more established in the field that we shouldn't even talk about, is it alive or not? We should talk about the degree of liveliness. And somehow, to me at least, and I don't know if this is entirely true, but it seems to be that degree of liveliness is correlated with complexity of the environment, negatively correlated, right? Inversely correlated. So the more complex the environment you need to sustain life, the less alive something is, the more. So a virus, for instance, I would put very low on the liveliness scale, right? Because it needs an extremely complex environment to self-replicate. Whereas something like us, we are kind of capable of living in, you know, we need less components, so to say, to maintain processes. Or let's not even talk about us. Maybe it's taking it a step too far. Let's talk about a cell, right? It requires a bit of a less complex environment, but it still requires an environment. There is no, as I said, there is no system without a larger system around it, right? Life does never happen in a vacuum. There's always an environment. And this is an important message, I think, when we talk about what is an autonomous system. So at the beginning, our synthetic cells will not be entirely autonomous. If that would be our ambition, maybe we should stop.